Wednesday morning here in sunny California. They say that engineering biology is hard, but audiovisual, it seems, is harder. I am joined here live with Will Canine from Opentrons, the founder and the chief product officer, and Kristin Ellis, who is the head of scientific development at Opentrons. And this is a sponsored webinar where you're going to learn all about the power of automation and how you can get one of these OT2s into your lab and into your life. The title of our SynBioBetaLive is to let 1,000 foundries bloom, and we are democratizing discovery in the age of synthetic biology. It's been a power week for synthetic biology, and I'm going to give you some of the latest breaking news that we put in the newsletter yesterday. And we want to hear your questions and comments. So we're live on YouTube. You can put your comments in the comment field below. You can tweet us, SynBioBeta, at SynBioBeta, or you can email us live@synbiobeta.com. Or if you want to just buy an Opentrons, you can go to info@opentrons.com and you can send them an email, info@opentrons.com. We're giving away a free ticket to SynBioBeta this October for the best question that we get. But first, here's the news of this week. 2018 is the year of cell-free synthetic biology, and Sutro Biopharma just announced a record $85.4 million Series E to fund novel cancer treatments. And Mammoth Biosciences just this week announced $23 million in funding for CRISPR. This week in the newsletter, we highlighted Pivot Bio and Karsten Temme, who is making probiotics for plants so that they can fix their own nitrogen, and they demonstrated some of the pictures of the crops doing field trials. And also Oxford Genetics and Ryan Carwood signed a multi-million-pound supply and licensing agreement for CRISPR for engineering mammalian cell lines. So some very exciting news that was going on. Next week we have even more exciting live video coverage, because I'm going to be live at Northwestern University, joined by Amy Trejo from Procter & Gamble. And we've got a special webinar format where we're going to be talking with Amy about how startups and anybody in the synthetic biology industry can partner with Procter & Gamble and some of the exciting projects that they have going on. And in 2 weeks' time, we're going to be launching a new webinar with my friend and colleague Mark Bunger from Innovation Lab. And we're going to be launching a brand new report on the growth of chemicals and materials in the synthetic biology industry. So we'll hope you'll join us for that as well. If you are watching at home, then we want you to send us a note in the YouTube comments, and we'll do shout outs to some of the many companies and individuals and organizations around the world who are joining us live. I wanted to remind you all that you are guinea pigs. In particular, Will and Kristin are the guinea pigs on the show today. This is the second SynBioBetaLive that we've done, and we're very happy for their support. And also, in particular, the nature of this we're modeling on Wayne's World here in the studio, so that we can have a live studio audience who is here with us today. Completely excited live studio audience going on there today. So we're relying on you and your questions to come in from the audience. So, Will, I always butcher your last name. It's not Canine, it's Canine.
Nice one.
Thank you.
Yes.
So I'm sorry for butchering your last name, but now you get to redeem yourselves and tell us a little bit about Opentrons. I mean, what is Opentrons? What's the OT2? And what was your motivation in starting the company?
Sure. Well, that's a big question, but I'll start with kind of the elevator pitch. Opentrons makes robots for biologists. A lot of you in the audience have probably spent time moving tiny amounts of liquid around from vial to vial to run your experiments. And you would not be alone. In fact, 90% of labs out there don't have access to any lab automation at all. That's what we're trying to fix. Our machine is an order of magnitude more affordable than the next comparable liquid handling station, starting at $4,000. And it's also fully open source, so you can write directly to our Python API or even modify the platform to suit your needs, as we'll see folks have done later on in our talk.
So when you think about how the OT2 works and about what your vision is, what's the impact that you hope to have? And I know in the past when you've talked about this at SynBioBeta, you've compared the OT2 to the desktop, where you've got companies like Cultured Robotics or Transcriptic or Emerald Cloud Lab. They're doing the mainframe. You think there's a need for the OT2 as a desktop machine on every bench? Is that right?
Yeah, I mean, so if you look at the history of computers, right, you started with the mainframe. And this was when one central facility would have a computer and people would sort of come to that facility to run different jobs. And that's really where liquid handling automation has been for the last 30 years since its invention, is these big, expensive, difficult to use, centralized automation platforms. And like what we saw with the computer revolution, with both first the PC, so the desktop personal computer coming out and really democratizing access to computation to every office and every home. And then we also saw the rise of the cloud computers, making it so that you could have access to big mainframe-like services from your own workstation at home. And we're seeing the same evolution now in labs. As you mentioned, there are things like Transcriptic, Emerald, Cultural Robotics, and all your traditional CROs really are all what you could call cloud labs, where you outsource your work to somebody else. And we are trying to make the PC of life science so that everybody can have automation on their bench. And if you ask, like, why do that? Why do we care? I really think that if you look at the big existential problems that we are facing as a species, like how are we going to feed 10 billion people? How are we going to make sure they don't all die of diseases? How are we going to scale up our manufacturing output and raise standards of living globally while shrinking our ecological input? Well, all of those are biological questions. They all have biotech answers. And right now, we're not making biotech fast enough. So we need to make automation accessible to 10 times more people so that they can all go out and make the important solutions that we need so desperately as humans.
Great. So we're going to show a couple of videos here. The first one is kind of like a marketing overview of the OT2, and then we're going to delve a little bit deeper into a technical video of what it can do. So let's run that first video.
A lot of work goes into a new discovery. Writing papers, preparing presentations, reading articles, designing experiments, analyzing data, not to mention lab work. So much repetitive pipetting goes into each discovery, and that's what Opentrons is for, getting your lab work done. This is the OT2, a personal lab robot that takes care of repetitive pipetting accurately and quickly. The OT2 transfers liquid using robotic pipettes that meet or exceed the accuracy and precision of other lab robots. Using high-performance parts and cutting-edge mechanical design techniques, the OT2 is faster than many robots 10 times more expensive. And you can use most standard labware and consumables, just like if you were running your experiments yourself by hand. Except you don't have to run it yourself. With the OT2, you can run a protocol from the other room over Wi-Fi. You can check on the progress of a job remotely and queue up more protocols to run. You can choose one of the protocols developed by our community and submitted from institutions like Stanford and the Mayo Clinic, or make your own automated protocol with the protocol designer. The intuitive visual interface makes it easy to create protocols yourself. Once you have exactly the protocol you want, you can simply send it to the robot to be run. And it's easy to set up a new run. All you need to do is remove your samples from the previous run and put on the new reagents and consumables, securing them easily on the precision-made deck. You've got plenty of work to do. Why not let Opentrons handle some of it for you? Get an OT2 pipetting robot for your lab, starting at $4,000.
All right, well, that's a cool video. That was filmed, we were talking just in the break, at Jenn Space in New York City. Can you tell us a little bit about how you got your start at Jenn Space?
Yeah. Jenn Space, for those of you who don't know, is the biohacker space in Brooklyn. And they have classes like the Biohacker Bootcamp or Intro to Synthetic Biology. And I went there to basically learn how to pipette, learn how to actually use these technologies. And I wasn't long into my first pipetting exercise when I was like, why can't I just download this protocol and hit run like I can on my 3D printer? And really that was the motivation that led me to meet my co-founder Chu, and brought us all to the place that we are today, where we're trying to eliminate manual pipetting. But I just think it's important to call out that we got our start in a very non-traditional space, but we do have a lot of traditional scientists like Kristin here, who's spent a lot of time pipetting. So I think that there's a picture that you can see of Kristin pipetting. Sorry about this. Do magic of audio visual.
So what we talked about in the past was also some of the start that you got both at Jenn Space and then also going into Y Combinator. So and now you're starting to see traction both with large biopharma companies getting these robots, in chemicals and materials companies. But Kristin, maybe you can tell us a little bit about your startup at the MD Arthur Anderson Center in Texas, and what you were working on there, and what drove you then to be excited about what Opentrons is doing.
Right. I was so I was actually doing a lot more pipetting than Will did. My background is a little more traditional in biochemistry and molecular biology. And when I was at MD Anderson in Texas, I was working on coordinating and running clinical research trials in pancreatic cancer. And I spent half of my time talking to patients, explaining to them the research that we were doing, recruiting them to trials, and the other half of my time I spent pipetting constantly, and running sort of the samples that we got from those patients as efficiently as I could, as a person sitting at the bench pipetting all day, but still not the most efficient use of my time. And when I discovered Opentrons, I was like, where has this been my whole life? We, even at MD Anderson, which is a very well-funded institution, individual lab budgets were pretty small, so it's not like I could go to my PI and say, hey man, buy me a robot so I can do more work. It was just, no, you just need to stay longer and do more work and still not be able to meet the throughput of a robot. So what really sparked my interest in Opentrons was the idea that we could put one of those on every bench and accelerate the research of every individual person, be they a grad student, a technician, a trial coordinator, that's sitting at the bench trying to push the forefront of cutting edge science in all of these places. And so, I sort of left my job at the bench and moved to Opentrons because I felt that, I would be able to have a bigger impact on the field, bringing this to fruition and bringing it to life.
So we talked a little bit about your founding story, Will, and about going through Y Combinator, getting funding from Coastal Ventures recently. And part of that's enabled you to hire expertise. And one of the experts that we're going to hear from in a second is Taylor Goodman. Taylor sounds like a special kind of guy. Where was he working at before Opentrons?
Well, Taylor was at MakerBot before. And just to give a little bit more context here, so those of you in the audience who have been to a SynBioBeta conference will have seen the OT1 robot. And that's our version 1 and what any lean startup would call the MVP, the minimum viable product. It's an awesome hacker platform. So people at Stanford and the Mayo Clinic and other places who really are highly technical users and love to write Python and hack on hardware, they love that machine. But it's not the right machine for what we think of as a mainstream biologist who really wants to plug in a robot and just have it run their experiments. And that's what the OT2 is. And so that's really the evolution that we've come through as a company, using our minimum viable product, the OT1, which was all off the shelf and 3D printed parts because we didn't have any money to put into research and development and making custom parts at the beginning of our company. But then through selling the OT1 and getting a lot of great success from customers, that's what got Y Combinator interested, that's what got Coastal Ventures interested, and that's when we got the funding to really invest in our remarkable engineering team in Brooklyn that has developed the OT2. And as John, you were alluding to earlier, the way that we make our machine so affordable compared to all other lab robotics is that we are using the same technologies as your standard desktop 3D printers, your MakerBots, Ultimakers, etc. And we are just using the same platform basically and putting a pipette onto it. And so that's why it's so great that Taylor and so many other folks from MakerBot have come to work on Opentrons. And so this next video is him really describing the hardware decisions, the design decisions that we made in order to make the most affordable and high precision lab robot that's ever been sold before.
Awesome. Well, let's see what Taylor has to say.
Hi, I'm Taylor Goodman, Director of Hardware at Opentrons. I'm going to explain some of the hardware design decisions we made in the OT2 to create the most affordable, high-precision liquid handling robot ever sold. First, let's take a look at the overall gantry system. Our goal was to create a rigid and precise structure using affordable manufacturing techniques. The main structural components, including the gantry, the frame, and the bridge, are high-precision extruded and CNC'd aluminum, creating a rigid design that can be built at high scale and at a low price. Additional structural integrity comes from the two sheet metal side panels on either side of the machine. The precision aluminum deck is machined to a high tolerance and is removable to allow for additional configurations in the future. The robot is enclosed in clear acrylic and comes with covers for the vents in the floor if you'd like full enclosure. The door has a friction hinge to stay open when you are loading and unloading the deck, but for safety, the robot will not run if the door is open. Next, I want to tell you about our new robotic pipettes. The pipettes are the business end of the machine, and we took a lot of care to ensure accuracy and precision are comparable to machines 10 times more expensive than the OT2. You can easily install them into the machine with an Allen wrench we've included with the robot. The OT2 can hold 2 pipettes, single channel or 8 channel. It doesn't matter which side of the head the pipettes are mounted to, and you can use 2 multi-channels or 2 single channels at the same time if you prefer. Here's a look inside the pipette. We use a robust micro-stepping motor attached to a precision molded volume chamber. The volume chamber is a cylinder with a piston that converts the linear motion of the motor into the desired volumetric output of the tip attached below. The tip cones are made from a hard composite plastic designed to put on and eject hundreds of thousands of tips throughout its lifetime. You can learn a lot more about our pipettes, including the specific linear millimeter to microliter calculations for each volume range on our white papers found in the pipettes page. The on-deck tip probe is a unique feature to the OT2. The robot uses it to calibrate the different sizes and brands of tips biologists prefer in their lab. The switches for the tip probe are under the trash container. The robot uses the switches to measure the location and length of a tip on the pipette that has been installed. The simple design allows the robot to stay lightweight and flexible while maintaining a resolution of a fraction of a millimeter in the XYZ coordinates. Okay, that's all for this video. I hope this was helpful for you to better understand our new lab robot. If you have any more questions about the OT2 hardware, you can look at our documentation or chat with one of us online. Thanks so much for watching.
What capabilities are we seeing in the OT2 that weren't available in the OT1, Kristin?
So some of the things that are improvements in the OT2 are definitely our fully integrated electronic pipettes. So with the OT1, we put manual pipettes that were controlled by a stepper motor that replaces your thumb, basically actuating the pipette. We've now integrated that motor inside of the pipette itself, which gives us very fine and very precise control over the accuracy and precision of the OT2 as an instrument. It's also much faster and much easier to use. So one of the big pieces of customer feedback that we took in with the people who use the OT1 as we transitioned to the OT2 was to make the platform far more usable. And so the hardware is higher fit and finish, it's more precise. That makes the hardware slightly less flexible, even though it's still open source, but it makes it eminently more usable. So your average bench scientist coming up to the OT2 can figure out within an hour how to set it up, how to use it. You can download a shared protocol from our library, set everything up on the deck, and be able to hit run. One of the things that we've talked about a lot in making this instrument is sort of the idea that introducing automation to more benches, to more labs, will increase not only through this open source platform the shareability of these protocols, but therefore, if you have these machine readable protocols that you can share within labs and between labs, right, you can not only increase the number of hits on target, so the number of times that you can run a particular assay and get a particular data set out of it, but you can also share that protocol with another lab, and that can therefore increase the replicability and reproducibility of results across the entire field. And that's something that we're really angling for when making this very consistent, very precise new machine.
So that's a bit of a game changer. I think that we've seen for previous hardware it's proprietary, or the hardware isn't open access. So you've got an open piece of hardware, open piece of software, open source software, and then how many protocols have you got that people can download right now?
We have dozens on the library right now, and we're increasing every day. So one of the big things, right, as more and more people get onto the platform, we start to see more and more of a network effect. One person uploads a protocol, and everybody else that has a robot can have access to it. And the more and more of those protocols that we get, the more and more people join the platform, the more and more people we have testing and validating the protocols that we already have on the library that are already available, the better and stronger the platform becomes. So it's really the scientific community that's working on the platform that's actually strengthening that protocol library and that process and making the product better. And that's been true since day one. I mean, we're working with customers at all levels. So we have customers at 7 of the top 10 big pharma companies. We have scientists working in 90% of the top 50 research universities. Customers that the synthetic biology crowd would be familiar with, like Modern Meadow and Lygos. Later, we'll be talking to some people in Drew Indy's lab at Stanford, and we'll be speaking with Doug Dinsmore at the DAMP lab at Boston University. So in addition to, we had the opportunity to give away 10 free robots to IGEM teams this year in order to see what effect automation might have on the interlab reproducibility study there. So the more and more people we can get on platform, the more and more we have shareability and reproducibility between all layers of industry, academia, and community labs.
Excellent. And so let's go in a second to the Indy lab at Stanford University. We were down there recently and we interviewed Keone Gandel, who is the head of the Free Genes project there at Stanford. So let's learn about how Keone is using the Opentrons.
My goal in working with the Opentrons is to erase the need for mundane, basic lab work and enable people like me to be able to build synthetic cells from scratch. The Free Genes project is a project to make free, accessible, and reusable DNA for everyone, to give everyone access to this wonderful biotechnology of the future.
So from Keone how he's using the OT2. Maybe you can tell us a little bit more about the Free Genes project.
Yeah, so the Free Genes project really shares our same goals of increasing accessibility of this important, powerful technology. Basically, anybody, you all listening right now, can submit a gene, a DNA part that the Free Genes project will then make available. Twist Synthesis has generously donated a lot of DNA to this project, and after you submit your part and it goes through a safety review, they'll synthesize it and send it to the Indy lab at Stanford, who will then process it into a easy vector and make stabs of it that they can then ship to the whole world.
Excellent. Let's hear more from Keone about how he's using the OT2.
It would be extremely difficult and very error-prone to do the Free Genes project without an Opentrons, because humans have human error, and if you compile that and if you do more and more, people tend to mess up more and more. When we began the project, we did everything manually, and there was an unacceptable amount of failure due to human error. And by porting it to the Opentrons, not only is human error no longer a problem, but we also have freed up our time to do other things. To do 100 genes manually took about a whole day of planning and operating, just to build the genes from the synthesis plates, and that doesn't include, transforming those genes, which took another 2 or 3 hours for just 100. Picking colonies, which took another 2 or 3 hours, and then sending out for sequencing and getting sequencing data back was one of the hardest parts, since it took up to 4 hours of mind-bendingly boring work to make sure everything was verified properly. And so we've gotten that entire process down to about an hour of human time. We started using very low volumes, so with other lab robots, such as the Echo, you can get down to less than 0.5 microliter reaction sizes. With the Opentrons, we've gotten down to about 5 microliters, which is still very small, and we've half the cost of all of our reagents, and in some cases almost fourth our costs. And since the Opentrons are so cheap, we can simply deploy more for more processing, and since all the software is open source, that scaling of up or scaling down becomes very easy, so that our lab can create a ton of genes with many Opentrons, but a smaller lab can create just a few genes with an Opentrons, without having to invest in this huge piece of equipment.
So that's pretty interesting. You're managing to quantify the benefits that you're able to produce for the end customer, both in terms of the time that they're saving and in terms of the volume and the consumable costs. Is that the main value prop that you're able to go to people with?
Well, and then there's the third that he mentioned as well, which is preventing human error. And the one that he's going to talk about next is making it so that the innovations that the Indy lab makes can be easily transferable to the rest of the scientific community via the Opentrons platform.
Great, let's run that video.
The reason we use the Opentrons is for 2 reasons: because it's affordable and because we can develop open source software for it. When you want to use a robot in many labs, those labs have to have the robot. That's where affordability comes in. Any lab can afford to own an Opentrons robot. And then software is very important, because if you don't have open source software, it's very difficult to share your protocols and processes with your peers. Other companies have very closed down software stacks that force you to go through the company if you want to get a protocol or a process. Opentrons makes that open source, so you can share directly with your peers, which is far more efficient, and I believe the only scalable way to operate in the future.
And so maybe you can talk a little bit about the open source repository. I think you're using GitHub, is that right?
Yeah, all of our code is on GitHub. You can go see live right now exactly what our engineering team is working on. And there are 2 ways that plays out. One is kind of the core code of our product, which, as you'll see, the Indy Lab has developed quite a few things on top of it, even morphing our product into a colony picker. And then there's the protocols, which are also open source and stored on GitHub, that you can get to from both our GitHub repository and protocols.opentrons.com. But yeah, we can hear Keone talk about the colony picker.
Excellent.
At the Open Foundry, we've also developed a colony picker, which you take a picture of the plate that you want to pick colonies from, and our software will automatically find the colonies, and if you put it onto the deck of an Opentrons, will pick the colonies for you. Instead of having a $50,000 robot or a $100,000 robot that only does that one process, now you have an affordable liquid handling robot that does all of your processes and also does colony picking, making it a very wise investment.
Great, very wise investment indeed. So we also went to speak with Anton Jackson-Smith, who's in the Indy Lab as well. He's even modded the Opentrons to be Alexa enabled. Is that right?
Yeah, exactly. Anton worked with a group of undergraduates to create a bunch of different customizations on the Opentrons platform, including an integration with Alexa, so you can say, Alexa, tell Opentrons to plate my samples, and it will do exactly that. He's also worked with folks to make a computer vision, calibration, as well as some plate moving arms on the robot.
Let's hear what Anton had to say.
We were really trying to achieve 2 things. The first was, if we can increase the ability of robots to do things using software or using sort of simple open source components like 3D printed parts, then those parts become available everywhere. The software becomes available everywhere, so we can upgrade everybody in the world's equipment all at once, which was really exciting to us. And the second and more important thing was we felt that most robotics in the lab was really designed to do one thing 1,000 times. So if you've got a protocol really well established, then it's worth the investment to use a robot. Whereas what we found ourselves doing was often doing 1,000 things one time each, and it's difficult to program a robot to do that. So how can we make it more efficient, more effective to use robotics as part of your everyday lab workflow?
Excellent. And Anton really also talks about the use of freeing up people to do important work. So let's listen to what he says about that.
So one of the most frustrating things about bench work for me is that you can take these incredibly intelligent people with decades of education and have them going like this for 8 hours a day. So in terms of scientific accessibility, I think it frees up people to do much more important work, and then you can focus on the science while the robot does the basic bits and pieces.
So Kristin, maybe you can tell us a little bit about some of the top 10 applications that people are using the Opentrons for.
Yeah, definitely. So in synthetic biology, as it relates to synthetic biology, we have people in the Indie Lab doing assemblies and transformations and digests. One of the more common applications we get asked for is NGS library prep. And so it can do a very wide variety of different protocols, as Anton mentioned. And then there are people who are having it do workhorse protocols, so you have to do several PCR, qPCR preps a day. You need to do serial dilutions. You need to map plates, normalize your DNA. All of those things are possible on the Opentrons.
And if you were not using the Opentrons, then what other equipment, you know, how much would you be spending if you wanted a robot that could do all of this?
So most robots, I mean, one of the answers that I like to give to this question, right? It's like, what would the average lab be spending? And really, the answer to that question is the average lab wouldn't be spending money on automation at all, because there's nothing else that exists at a price point that's accessible enough for people to have access to it. But if you had a lab with a budget, your average lab with a budget that could actually do this, the next robot that's comparable, that can do the same things that the Opentrons can do, starts at probably $40,000,
wow.
And that's sort of your baseline.
Yeah.
And then it only goes up from there as you add functionality and capability. And let's not forget that a lot of these robots have to be programmed by automation engineers, and they do one thing very repeatably very well. With Opentrons and with our protocol library that we keep mentioning, you can do several things on the platform for a much lower reagent cost, a much lower capital equipment cost, and really have the flexibility and throughput to get a lot more done in your lab on automation.
Excellent. Let's hear what Anton has to say about his use of DNA assembly.
One of the fundamentals of biotechnology, bioengineering is just getting access to working DNA that is the sequence that you want. It's surprisingly hard to do that. And so at the moment, you can get clonal DNA from big synthesis companies like Twist and like IDT, but that's kind of expensive, and it limits your workflow, particularly if you're not in the United States. So Open Foundry lets anyone start building clonal DNA that's more complex than they used to be able to in their own lab. And the fact that it's open, I think, is really useful, because particularly if you're not in an academic environment, but if you want to start a company or if you're a DIY hackerspace, building a foundry from scratch is like the first thing any company working in this space has to do. They have to write all the software, they have to develop the instrumentation. And so we hope to enable like the next generation of hackerspaces and startups to be able to build whatever they want. The source code for Open Foundry is all online on our GitHub. The protocols and some documentation are there, and very soon there'll be a paper coming out that describes the Open Foundry model, how it works, and the combination of those things will basically let you buy a few simple tools, get the reagents, download the software, and get it up and running.
So there's some pretty interesting statistics that you shared with me earlier, Will. In particular, 3 out of 4 of your customers have never had a lab robot before. That's breaking new ground. That's creating new markets. That's pretty incredible.
Well, thanks. That's definitely what we're trying to achieve. And like Anton said, the first thing that any of these new bio companies have to do is create their own foundry. And previously that would involve a lot of engineering time, but with projects like the Open Foundry and Opentrons, now that foundry becomes an off-the-shelf part that these labs can just buy and install and start years ahead in their progress from where companies even 1 year ago could have started. So I think that stat that you just mentioned, that 3 out of 4 of our customers have never had a lab robot before, that's really a testament to how we're making these things more accessible, more easy to use, and off-the-shelf solutions.
And can you talk a little bit about the big pharma companies and what they're running?
Well, so yeah, we do have that 25% of our customer base that does already have big robots, but even the biggest pharmaceutical companies in the world, with all the money to spend on automation, still only like 75–80% of their work will be automated, and they have people who are trying to figure out how can they move that to 85–90% of their work being automated. And Opentrons ends up being the perfect solution for them because it's smaller scale, it's easier to use, so you don't have to use your valuable technical programming employees in order to use the Opentrons, and it fits in the niches where the bigger, more expensive robots are just too big and unwieldy.
And we talked a bit about IGEM earlier, so the genetically engineered machines competition. Kristin, what's Opentrons doing with IGEM to enable the next generation of bioengineers to use automation?
This is one of my favorite things to talk about. So we got the opportunity to partner with IGEM this year to basically give 10 robots to 10 different IGEM teams. So we put out a call for applications to IGEM teams all over the world and had them answer a few questions and tell us about how they wanted to use the robot. And we gave away 10 robots to IGEM teams, and they're active and they're using them now. One of my favorite stories to come out of this actually is the first IGEM team in Uganda now is actually also the first team to have access to lab automation, which is huge. And they're making great strides and have a very big growing synthetic biology community. They're able to do community events, introduce high school students in the area to robotics. Newcastle IGEM is running a lot of design of experiments and trying to look at how can you use automation to optimize things like measurement in synthetic biology. And these are not just projects that the IGEM teams are doing for IGEM. These are projects that could have an impact on the field of synthetic biology as a whole, and how we think about the ways that we measure different constructs and how we think about the ways that we assemble different constructs. And so introducing automation to these teams of undergraduates and high schoolers and graduate students, and sort of leveraging their sort of fresh look at how automation can be integrated into these workflows has been really, really exciting. And we have a lot more that we're going to share, of course, throughout the competition season, and we're really, really excited to see what these teams are able to do with this automation platform.
Excellent. You mentioned design of experiments. Can you go into a little bit more detail of that, about what that is and what they're using it for?
Yeah, of course. So this is something that we're actually going to be talking about in a minute with the DAMP lab as well, on a little bit of a bigger scale. But essentially, you're looking at sort of there are a few different pieces that you have to think about when you're designing an experiment. So you have the initial setup of the experiment. You have actually optimizing the run, which is the automation piece, the bits that you're actually moving around. Then you analyze the data, and then ideally in a fully optimized cycle, you're able to take that output, feed it back into the design, and optimize that experiment, run it again, and see what your new output is, and sort of tighten that build design, build, test loop in synthetic biology, and make that sort of a more concrete piece. And it enables us to approach synthetic biology as fundamentally a discipline of engineering, right? So it allows us to apply some of those same principles to biology and to engineering these biological constructs.
So you talked a bit about Doug Densmore at the DAMP lab. We're going to go over to the DAMP lab shortly. Can you tell us what the DAMP lab is?
Yes, so Doug does this a lot better than I do, but I'm going to try to do it justice. So DAMP stands for Design, Automation, Manufacturing, and Prototyping. What he says is it's not wet, it's not dry, it's damp. And so what they're doing is exactly what I just said. They're sort of integrating that engineering ethos into the entire biological design and experimentation process. So they want to be able to design and build and test over and over in iterative cycles through this sort of perfect intermarriage of design software, their automation layer, and then looking at their data, their output, and being able to tighten that loop and feed all of that back in. That enables them to optimize things like, you can start to look at over the course of time what your total reagent costs. You can actually more accurately predict what your reagent cost is going to be for a particular project and how to sort of decrease that iteratively as you go along. You can start to predict which constructs are actually going to work, what the output should look like if you combine things in a certain way. And you can really start to optimize around designing and building products and constructs that will actually predictively do the thing that you're predicting that they will do. This is something that the DAMP lab has actually done with other robotic systems. So they did this on a TCAN system, and now they're repeating the same process on Opentrons, because if you can take the automation piece of that, the automation infrastructure, and you can make that even less expensive, this becomes similar to the Foundry, almost a modular thing that you can, a modular paradigm that you can introduce to several different laboratory contexts and be able to implement that kind of robust engineering type thinking into the design of your experiments.
Great. So Will and I went down to Boston University last month and we interviewed Doug. So let's take a look. So tell us a little bit about this philosophy of Opentrons, kind of the way it works with a lot of other platforms. What other platforms are you integrated to?
What we need to do in my lab is we need to translate high-level biological descriptions into DNA.
Okay.
And along that process, we need to create lots of software tools.
Okay.
And if those software tools have to produce very rigid formats or can't communicate automatically or seamlessly with the hardware, it's very difficult to integrate them in our workflow.
Got it.
So with Opentrons, when I talked to Will, they had a very open philosophy, like this is how the code works, this is the hardware. If you want to talk with our engineers, we'll work with you to figure out what APIs might be needed, making sure that we could exchange data. So we knew that working with them was going to be really key to getting things integrated. So that was exciting to me.
So any thoughts on the DAMP lab and how Doug is using it?
Well, I think it's another great example of how when you have an open source platform, you just let people get started way more advanced, in a way more advanced position than if they didn't have all the stuff that already existed in Opentrons. Because previous to Opentrons, they had to do a lot of hacking of proprietary systems, building their own custom hardware. But now with Opentrons, they can just plug their software right into our open system and get up and running.
Excellent. So we asked Doug a little bit about how the Opentrons is changing the nature of how his scientists work. So let's listen to what he had to say. So if you go back before you had the OT2 in the lab, talk about the whole design of experiment and how it's changed because you've got this low-cost automation solution. For a typical grad student or postdoc in your lab, are you kind of changing the way that you teach because of this new capability that you have?
We change, yes. So what will happen with robotics is we'll change how students spend their day. So instead of looking at my day, getting up and saying, well, I have all of these experiments to do, it may be that I have to run this software, set up some robotics, and then I can now spend my time with a more complicated assay or more complicated characterization, or actually becoming interdisciplinary. So where before a biological student couldn't learn a programming language because they're busy in the lab, now they might learn about programming, they might learn about biophysics, they might learn about bioinformatics. So they can become they have more of a breadth of education. And I think that will make the kind of synthetic biology workforce we want to see.
And are you seeing that too in terms of, let's say, how IGEM is going to be using it? Is it changing the way that undergrads are doing science? Is it changing the way that they go about their first day in the lab?
Definitely. It's definitely doing that. I mean, we have several examples across our community of users, scientific members of our community that specifically are getting the Opentrons or several Opentrons in order to sort of smooth that entry into bench research. So we have one example, Piyul Rabani, who works at NYU, and she has 3-month rotation. She has students come in for 3-month rotations, and she also has a lab to run and a lot of data to produce. And so these students come in and they want to be able to learn what biology research is all about, how to apply the tools of molecular biology to a problem. And often before the OT2, instead of actually, learning the sort of higher level, what is a PCR and what can it do and why do I need it, they're learning how to mix the different liquids very precisely by hand and doing that over and over and over for several weeks until they get good enough at pipetting a PCR to then make the data and then talk about what the data means. Now we have been able to introduce the OT2 to her lab. She has been able to pull together several different scripts to be able to run these PCRs, and so she can walk in and tell the students, okay, here's how you set up the PCR, and then dive right into why is that a meaningful assay, what does it mean, how does it apply to our research, what data are we looking for, how do we design an experiment and pick the assays that are going to give us the data that we're looking for, and spend a lot less time over that 3-month period coaching those students on how to pipette very precisely, which robots are much better at, but the students are going to be much better at imagining and thinking about, what sort of experiments can we run. Paul Jensen at the University of Illinois is also purchased 3 OT2s and is running a pilot program where he wants to be able to teach biology students how to code. So he wants to introduce them to this open source platform and be able to combine concepts of programming and biology and designing experiments with this automation paradigm. And so through this pilot program, what he's hoping to do is fund a bigger grant, a continuing education grant that will start to change the way that we think about designing and doing biology experiments from the word go when students are entering the biology lab as undergraduates. So there's a lot of potential in changing the way that we think about how to work in a biology lab and conceptualize the way that we do bench work.
Great. And Doug also had some thoughts on the right kinds of projects and goals that he thought were good for getting started on the Opentrons. So as an esteemed professor at BU, I'm sure that you don't actually touch any of the equipment.
No, that is true.
But what are the top 3 things that the machine's being used for in your lab?
So the things that we really want to automate, one of the things we want to do with assembly, restriction enzyme-based cloning methods. Then we want to automate transformation and also things like mini prep, kind of basic routines that we would do in the lab. Okay. So what we need to do, of course, is, again, as I have pointed out when I talk to folks, is think about which ones can really benefit from a robotic automation. It's definitely not, I think anyone would be disingenuous to say we're going to automate everything on the robot. That's not appropriate. It's not only not appropriate, but it's not the best use of the resources. Yeah. So really thinking selectively in terms of cost, time, accuracy of results, and that's kind of how we evaluate what to automate first.
So one of the things that we talked with Doug about was this idea of a synthetic biology stack, and this is something that we've been talking about within the industry for quite a long time. There's about 520 companies now in the synthetic biology industry, and we use this model of the synthetic biology stack, and you can see it on the screen here, starting off at the base with the gene and genome synthesis and sequencing, so reading and writing of DNA. Next up, you have this tools layer. This is where Opentrons would sit. On the automation side, you've also got cloud labs there, and you've got BioCAD tools for computer-aided design of genes and genomes. Next up, you have organism engineering platforms, that is, companies that are making organisms that could be used for many different applications. And then at the top, you have the application stack. This could be a company that's making a material or a chemical or a food product. So maybe you can talk a little bit about where Opentrons fits in that automation layer and where you see the shape of the industry changing over time.
Yeah, absolutely. I think that the stack that you all have is the full synthetic biology stack, but when we really think about it, we're thinking about that tools layer that sort of starts at the top with software that allows people to easily design their biological parts and design their experiments. And then that layer needs to feed into an execution layer, be it a cloud lab or a person pipetting by hand or a lab robot sitting in your bench. And the thing that we really try to do at Opentrons as an open platform is say, this software, this software, this software, it doesn't matter, because we're open, all of them can pipe into our platform. And so the execution layer is agnostic to what is above and below it in that stack. And that's a really important part for continuing this ecosystem to grow and evolve, is making sure that these pieces are modular and can be swapped out as people need.
Great. And maybe you can tell us a little bit more, Will, about the product roadmap, both for hardware for the OT2. What does the OT3 look like? Maybe then tell us about the software as well.
Sure, yeah, there will always be an OT3, and we welcome any suggestions from the audience. We love hearing those. So far, the best one that I've heard is the quadcopter with a pipette on it, just flying around and running your experiments in the lab. Let me know if you guys like that idea. We might make it real, but probably not. But seriously, what we're considering doing is, and what we are doing, is putting modules on the base of the robot. So, pipetting is by far the most common activity, and it's what strings together all the different parts of a protocol. But you also need to do things like heat and cool your reagents. And so we have the temperature deck that will hold your reagents a specific temperature from 4 degrees up to 100 degrees Celsius. You also might need to do things like separate out your nucleic acids from the rest of your cellular detritus, or pick out that specific protein with a his tag on it. And so for doing that, we have our magnet module, which will engage and disengage magnets to your labware to do magnetic bead-based purifications. And so those are the two pieces of hardware that are coming out very, very soon.
Excellent. And in terms of software?
Oh, in terms of software, so today the only way to create your protocols on Opentrons is to use our Python API and write code. A lot of people really love that, but it does add a barrier of entry for the more mainstream biologists out there, the folks that really just want the machine to plug in and work for them. And for that set of users, we've been developing a visual protocol designer that allows you to, in your browser, click around and drag a 96 well plate on, say what reagents are in it, add a transfer command, and visually produce your protocol that way. And so that's in beta test right now, and it's going to be live within the next few weeks.
Great. So we moved on to the Q&A part of the show. We have a couple of questions that have come in from people online. The first is, do you have to calibrate the Opentrons machine after each use?
So no, you don't have to calibrate the Opentrons machine after each use. This is another one of those things that's an improvement between the OT1 and the OT2 that we love talking about. So there are a few different types of calibration that we talk about on the OT2. We have our deck calibration, and so that is just the basic calibration you do it once, right after you pull it out of the box, and that just ensures that nothing got misaligned during shipping. Then you have your tip probe, which Taylor described in our hardware video, and that is how the robot recognizes the dimensions of whatever tips you have placed on the deck. Then when you upload a protocol, you have the opportunity to go through our calibration workflow there. And so what the robot will do is it will pick up a tip and it will go to the first well in whatever plate or tube rack you have on the deck, and you'll be able to confirm or jog around just slightly to sort of refine that calibration and then hit save. Once you do that for a protocol that you want to run, when you come back and re-upload that protocol, or if you choose to restart that protocol after you've run it once, you should not need to go through that calibration process again. So you should be able to do it once, just go through, check, make sure everything looks the way you want it to look, and then you should be able to run that repeatedly as many times as you need to. And we've left it flexible so you can go back and double check your calibration as much or as little as you want to. We kind of want to leave that in the user's hands. I know that I'm superstitious, so I might have an inclination to go back and double check more than once, but I mean, for our robots, I usually don't. I just let it run several times.
Great. And another question we had was, do you need any specific equipment, in particular tips for use on the OT2?
No, you do not.
Go for it.
We make it open to whatever type of tips you need, as long as they're universal bore. We do sell tips that are optimized for automation, but you can use the tips that you already have in your lab, as long as they don't use a proprietary type of connector to the pipette. And also in terms of labware, we get that question a lot. Do you need specific proprietary labware? The answer is, again, no. Our goal is to make this automation platform accessible to all labs, no matter what type of plate or tube you're using. And so we've gone through great pains to make it possible for you to use whatever kind of funky container you want, as well as the tried and true 96 PCR plates.
Excellent. Kevin, do we have any other questions from our listeners online?
Yeah, there's a question about the software development activities. How can people participate or recommend either new protocols or features in the software platform?
I love this question.
Yeah, this is
Thank
a great
you for
one.
this question. So there are a couple of answers to it. So first, because we do all of our development on GitHub, you can go and look at the current sprint that has been planned right now that our engineers are doing. There are tickets on there that people are assigned to, and you can be like, oh, I see that the feature that I'm interested in is being developed right now, and I really want to make sure that this little detail is taken care of, so I can add a comment to the ticket on GitHub and provide that information directly to the engineer that is coding up that ticket. You can also submit a pull request. We love pull requests. So if you want to make, the keyboard shortcuts on the OT1 app was actually a pull request that one of our awesome users in Seattle developed and submitted to us. So we love it when people will give us new features that we can just merge right in. And then third, in terms of a protocol, lots and lots of different ways that we can do that. One is, if you create a protocol that you want to add to the library, again, just submit a pull request and we'll put it on there. If there's a protocol that you want that isn't there yet, just chat to us online. There's a little chat widget on our website and on in our app, and we're also active on Twitter, so you can just ask us, hey, can you create this protocol for us? And we'll be like, yeah, we're actually doing the same thing for a customer right now. What's the input that you would like to have? So, the short answer is, any way that you think you can submit, we would love to hear from you.
Excellent. We have another question from online.
Right, yeah. The last question that we received was, what is planned for OT3? What are the hardware improvements that you're thinking about right now?
Well, yeah, good question. I'll try and give a bit of a more serious answer than my quadcopter answer. So we're about 3 months into the OT2, so we're still definitely learning the things that work really well and the things that could still be improved upon on the OT2. So I imagine there will be some small things in terms of, optimizing how the robot moves and how we can pack and ship it and things like that. We are also thinking about how to make the platform bigger so that you can fit even more samples on the deck and larger volume ranges of the pipette. And we're also thinking about how we can make the platform sterile. So we have a version of our OT1 robot that'll fit inside of a sterile tissue culture hood. And we have a lot of people who really love that feature. And we are figuring out how to provide that sterile environment on the OT2 as well. And that will either be by making a system of the OT2 that can fit into a hood, or by putting a HEPA filter on top of the robot. And so we'd love to hear from you which of those two solutions you prefer for your own sterile tissue culture automation needs.
Great, excellent. Well, if we have any questions, then type them into the box below, and we'll try to get to them in the next couple of minutes. I wanted to think about the future of the company, and you've got manufacturing base in Shenzhen in China. You've got headquarters in New York. What are you thinking about? Do you think that's where you'll be? You're thinking about setting up a Bay Area presence?
Well, we are always in the Bay Area, traveling or at a SynBioBeta or another conference, and we've got a ton of great super users in the area who are more than happy to chat with folks that are interested or show off a robot or anything like that. So we are not formally in the Bay Area yet, but we do have a lot of informal presence here if people would like to reach out. And we love being based in New York. It gives us a lot more access to our European customers also, which is really great. But yeah, as we continue to grow, and we have a robot on almost every continent now, including a boat on its way down to Antarctica.
Wow.
So we are always expanding, and we'll continue to do so. And we love sunny California just like everybody else. So.
Are you on the space station yet?
No.
Oh, but fun fact, actually, IGEM. I love talking about this. Sorry, but so the team from Queens, Canada, is also working with the Queens Reduced Gravity Experimentation team to do some crazy genes and space stuff that I'm not entirely up to speed on yet. But so I just keep in my head, I'm like, okay, yes, that's a possibility. We might get it on the space station. That might not be real, but genes in space.
Awesome. Well, if we don't have any other questions from the audience, then I'm going to wrap up. It's a pleasure to have had you all join us for this SynBioBetaLive. Thank you to Will Canine,
nice one,
the Chief Product Officer and co-founder of Opentrons, and to Kristin Ellis, the Head of Scientific Development. Thanks also to our friends at Stanford for Drew Endy's lab, Keone Gandel, Anton Jackson Smith, and also thanks to Boston University and Doug Densmore. And thanks to the SynBioBeta team and Garrett Kushner for putting this all together. We hope that you'll join us next week for a webinar with Procter & Gamble. That's on August the 14th at 9 a.m. Pacific or 11 a.m. Central Time in Chicago. And I also wanted to tell you about something very exciting called Synthetic Biology Week, which we're about to launch tomorrow morning. And these are a whole series of free satellite events that are taking place the week of SynBioBeta here in the Bay Area. So I hope that you'll come join us, and thanks to our sponsors for Synthetic Biology Week as well. Also, watch out for our newsletter. You can sign up at synbiobeta.com. We're going to be putting out our first report on chemicals and materials coming up soon. And register for SynBioBeta itself before the end of August if you want the early bird discount. So I want to end by thanking all of you for joining us, thanking again Opentrons for sponsoring this. And as I always say, may all of your DNA dreams come true. Thank you so much.
Thank you.